Hello everyone, my name is Yuzhen Ye. I'm a professor in the computer science department at Indiana University, Bloomington. Today it's my great pleasure to introduce some of our work concerning microbiome. In the past many years, we have been developing computational methods and algorithms to utilize multi-omics data of microbial communities and their networks to help us understand the composition and the functions of those microbial organisms in different environments. And also I will show a little bit about our work utilizing those bacterial immune systems, so-called CRISPR-Cas systems, to help us understand the microbiome. So by now, probably everyone knows microbiomes, they're important. There are good ones and the bad ones out there, but a lot of them are good. They play important roles in the health of humans, animals, and pretty much every environment. And in the age of precision medicine and precision health, we not only need to consider human genomes, but also we need to look into the human second genome. That's the collective genome of all the bacterial species that live on inside of our bodies. Let me first introduce some terminologies and concepts. Microbiome refers to the community of microorganisms, can be fungi, bacteria, and viruses that exist in a particular environment, such as human gut. And sometimes, if people really want to focus on microorganisms themselves, they use microbiota, and people also use metagenome if we refer to a mixture of genomes of the microbial organisms in an environment. One concept that's important is the MAGs, so-called metagenome assembled genomes. Unlike the traditional way of getting individual genomes of individual species, microbial organisms can be reconstructed from metagenome data. With a collection of metagenome data of the same environment, we may be able to reconstruct dozens, hundreds, and even more of those MAGs from those metagenome data. People have been looking into genomics data or transcriptomics data or metapromics data of those various of microbial communities. And if we do metagenomics from a collection of microorganisms, we can get a bunch of short reads. If we do shotgun metagenomics, we can get back a lot of short reads. And depending on your purpose, you may just look at those short reads as a bag of reads. You don't care where they're from, which species, and you can just treat them as a bag of reads. Sometimes you can do a summary of those reads. You get a bag of genes, predict from those context, and from those genes you may reconstruct the pathways representing the functionality of a whole microbial community. And if you have many metagenome data, you can do assembly and bidding so that you can separate those contexts from the assembly into different groups, hoping that each group of contexts represents a species. And then with those MAGs, bags of genes, we can do some interesting work. People have looked into microbiomes associated with different environments. The most famous microbiome project probably is the human microbiome project that looks into the microbiomes of healthy human individuals and also individuals with different diseases. There are also studies that look into the ocean microbiome and soil microbiome and so on. For us, we did something fun last year or this year as part of the Ladi summer camp for high school students. The goal of this summer camp is really to attract high school students so that they may get interested in technology and science. As a part of this pulp microbiome project, we did everything in 5 days, starting with collecting water samples from the neighborhood ponds and the lakes in Bloomington. And we have the DNA extracted from those water samples and have those DNA sequenced. in genomic sequencing center on IU campus. And from once we have the reads, we have the students look into the reads and did some analysis of the reads, and from there to analyze, to do some comparison of the microbiome associated with different ponds and lakes and so on. It was a fun project. Over the years, we have developed quite some algorithm tools for microbiome research with different purposes. The early ones, we focus on learning the basic stuff to do prediction from short reads or context. We come up with faster algorithm for similarity search, like rep search too, although there are even faster ones developed by other research groups. And we have methods to reconstruct biological pathways for metagenomes, and we do utilize how we can utilize those metagenomic sequences for function notation and so on. We also have been developing some methods for some interesting genomic elements, like those diversity generating rich elements. Those elements generate those hypervariations in the target genes, and also we have been looking into CRISPR-Cas systems. So we have given some new methods or targeted methods for characterizing and utilization of those systems. And we have been looking into ways to do integrative analysis or to utilize multi-omics data to help us to understand the microbiome communities. And also we have been looking to other direction to utilize those microbiome data to build like predictive models that can be used to predict human host phenotypes based on microbiome. Different omics data, including genomics, transcriptomics, and proteomics, they do provide complementary information that will be useful for understanding the functionality of microbial communities. Although out there, genomics data is more prevalent than the others for microbiome research, but the other omics data are technologies are coming along. In the recent years, we have been developing methods that can be used to analyze those metaproteomics data in a couple of different directions. I will introduce some of those methods we have developed. For one of our efforts to utilize those genomic data or metagenome data is really we have been trying to develop methods to characterize those microbial marker genes that are important for understanding human host phenotypes, and also they can be used to construct those predict models for host phenotype prediction. So the idea behind our subject example approach that's different from the typical comparative analysis, and really we kind of do the comparison step. We move this comparison step afterwards. The typical traditional way of doing comparative analysis is really we have a lot of shotgun metagenomic sequences. We do assembly, get conics, assemble, predict genes from them, and hopefully from there we can do comparison between the two groups of samples. We can get differential genes or functions or biological pathways, and from there we can utilize those differential microbial markers. Our subjective approaches, we kind of move up this subjective or comparison step, because in the end, starting from a large collection of short reads, we end up with really just a small collection of those differential genes. So we do this subjective step so that we will only, with the hope of getting only those differential reads that are likely from those differential genes. So the subjective step is based on the identification of those differential k-mers, and once we have those differential k-mers, they're either unique to a group of samples, or they're more abundant or less abundant in one group versus the other. So from those differential reads, we can hopefully just focus on those differential rates and from there get context and those genes. Recent work we have, recent method we have developed along this line is those locality sensitive hashing based k-mer clustering for identification of differential microbiome markers. So the idea is really, instead of working with individual k-mers, we cluster k-mers into co-abundance groups, so that from billions of k-mers that are typically contained, found in like a cohort of data sets, we have millions of k-mers. We apply this locality sensitive hashing algorithm so that we can group them into clusters of k-mers, and each cluster of k-mers contain k-mers that share similar abundance profiles across different samples. So once we have the clusters, we can do statistical testing on the clusters, and then we can find those differential clusters of k-mers that are likely from those differential genes. So we show really by adding that clustering step, we can actually improve this assembly of those differential genomes as the test on using those simulated data sets with 10 genomes, different genomes on the left. And also we show when apply this method to real data sets, it's likely that we're getting pretty good collection of those microbiome markers that can be used to build promising predictors for human host phenotype prediction based on microbiome data. A different microbiome data we have been looking into is metaproteomics data. Basically what we're looking at is a collection of these spectrum that were produced by mass spectrometry, and behind each of those spectrum, each spectrum represents a potential peptide that can be found in the proteins expressed in a microbial communities. So once we can identify those peptides, from spectrum, we can look into the proteins and their abundances in microbial communities. So the hope is those protein information can provide more direct functional insights into those microbial communities. But we know that peptide protein identification from this metaproteomic and tandem mass data is challenging because those data are produced from a complex protein mixture, and there can be a lot of proteins expressed by similar species or quite divergent species, and also there can be like different isolates or different strains from the same species, and comparing to proteomics data from individual species, the dynamic range of the abundance is even bigger for those community level proteomics data, because the species, they are of different abundances, and of course on top of that there will be expression differences of different proteins in the same species. So traditionally there are two ways of doing peptide and protein identification from proteomic data. One approach is really to utilize those reference protein database from which we can produce those theoretical spectrum, and we can match the real experimental spectrum against those theoretical spectrum, so that we can find the good matches, and from there we can identify potential peptides. So for this approach to work, we rely on a good collection of reference proteins, so that can be inferred from reference genomes, or in the case of microbiome, those proteins can be inferred from those metagenomes. But most likely, the reference metagenomes are incomplete, and they can be inaccurate because a lot of those genomes are assembled from metagenome data, so they might be inaccurate. But even though those reference metagenomes are huge, they are still incomplete. So it's not really uncommon that for a metaproteomics data, less than like 10% of those tandem mass spectrum can be identified. If we do have the metagenome available for the analysis of metagenomic data, that can really help. So if we have also that works if we have a matched metatranscriptomic data. So if we can assemble those metagenomic and or metatranscriptomic data into context, and from there we can generate reference protein database for peptide identification. The problem there is usually the assemblies of metagenomes won't be very good because of this ambiguity among the fragments. Good thing is really most metagenomes assemblers, they will produce those assembly graphs. So those assembly graphs will contain this overlapping ambiguity among the fragments. The problem is how we can utilize those graphs for metaproteomics data analysis. So one of our approach we call Graph2 Pro algorithm is really based on a graph traversal in those assembly graphs, so that we can produce potential peptides from those assembly graphs, and then from those peptides we can predict their theoretical spectrum. We do spectrum matching so that we can identify what are the likely peptides behind the spectrum. So once the peptides can identify, we can map them back to the assembly graph. We can extend those peptides. Hopefully we can get bigger fragments or even whole length proteins. So we tested our approaches on this wastewater metaproteomics data set. The reason we used that data set was really the identification ratio was very low for this wastewater metaproteomics data. If you just use context from the samplers, you predict genes and the peptides from those genes, you can identify like less than 10% of the spectrum. But if we can utilize this assembly graph, we show that we can dramatically improve the identification of this spectrum, although the ratio is still pretty low for this wastewater microbiome data. And the other idea is really, can we utilize this, fully utilize the genomic sequences information that may capture the variations of the genomes that are not in the assembly graph. So that's the idea behind the VR to PEP approach. So once we have the assembly graph, we can identify those peptides and proteins from those assembly graph that have matching spectrum, and from there we can find genomic fragments that may contain similar peptides with those identified from Graph2 Pro or just using the context. And by incorporating those variations in the genomic sequences, we show that we can further improve the identification of metaproteomics data. And you can see the improvement is pretty big, significant, comparing the red bars that shows the number of peptides that can identify from the spectrum data when we use the Graph2 Pro approach, plus this approach that considers the variations in the genomic sequences. And you can see that improvement is consistently found across all the data sets of this wastewater cohort. In recent years, because of the accumulation of genome, bacterial or microbial genomes, and also metagenomes, it's becoming possible to use a universal reference database for metaproximate data analysis, especially for of those data from well-studied environments such as human gut. So the problem is that the reference database is huge, contains like thousands of genomes. that translates into millions of proteins. So if we just do database search against these millions of proteins, the search won't be effective, and also it's very tricky to control the false discovery rate. Because of those reasons, we developed HiTSeq, which uses a two-steps approach to optimize the use of reference genomes or those metagenome assembled genomes. So the first step is to simply search experimental spectrum against the theoretical spectrum from high abundance proteins, so that the search database is small. From the search results, we can profile, we can decide on what species we need to include for those expanded database search, in which include all the genes, all the proteins encoded by those selected genomes. We show that by using these two steps approach, we can HiTSeq can provide a pretty accurate and also fast identification of peptides from metaproteomics data. The test on this synthetic metaproteomics data that was produced from a genome called Sisumi dataset. So HiTSeq was able to identify all 5 abundant species in this cohort. The other 3 species are very rare, and also we show that we cannot be too aggressive to identify the species, otherwise we're going to include a lot of false identifications. We also applied HiTSeq to identify peptides and proteins from metaproteomics data derived from real samples. We'll compare the results from HiTSeq to the results from MetaPro-IQ, another state-of-the-art peptide identification software, and also we compared the results with the identification when matched metagenome approach was used. So the results show that HiTSeq can identify more peptides comparing to MetaPro-IQ. At the same time, it's faster, and actually the identification rate using HiTSeq actually is even more than the identification when matched metagenome was used for the identification of peptides. So with HiTSeq developed, we applied that approach to more than 1,000 metaproteomics datasets, hoping that we can learn about the protein landscape of those important human gut bacterial species. We know that metaproteomics throughput is still low comparing to metagenomics and metatranscriptomics, but by incorporating data from more than 1,000 metaproteomics data, we can get a pretty good survey of the expression for like 100, for example, genomes. And we show that the top 100 genomes, that's just less than 4% of the total number of genomes that have some kind of expression detected in some of those data sets. But only this, but if we only look at these 100 genomes, they can explain more than 60% of the spectrum, identified spectrum, and those genomes are from 34 genus, and also they're from 5 of this most abundant phylum. And we can look into the individual species. We see there are some species that are found everywhere in many different samples, samples from healthy individuals or samples from patients with different diseases. And also we call those species generalists. We also found some species that are highly expressed, but only in a much smaller, more focused list of samples. So they might be more related to some diseases and so on. And we do show that these specialists and generalists, they do have different functional profiles of those proteins expressed. So we just show a couple of those examples here. For example, the functions, the proteins encoding for the functions in the carbohydrate transfer and the metabolism, they're more abundantly expressed in those specialists. And also the journalists, they do have more proteins expressed in those in some of those other functional categories. So the hope is really by using beta-proteomics data sets from many samples, we can learn the protein landscapes of some of those important human gut bacteria. That strategy of using the universal reference genome for database search-based metaproteomics data analysis will work for some of those well-studied environments, microbiome, such as human gut microbiome. Probably that approach will not work for those less well-studied environments. So, but we do have the other strategy. Can we just look at the spectrum? Can we de novo sequence those peptides? So de novo peptide sequencing, the idea was not new. It was out there for a little while, but the problem with this de novo peptide sequencing was that really the peptides were not very accurate. So that's the reason that the more commonly used approach for analyzing proteomics or metaproteomics data is to do reference-based search. But due to the recent advances of those deep learning approaches, we show really we can develop a method for de novo peptide sequencing that can give pretty accurate peptides. So our PepNet approach produces peptides that can have their positional accuracy is more than 90%. So that's a pretty encouraging performance comparing to what can be achieved by the current other two also deep learning based approaches. So PepNet is based on a fully convolutional neural network, a pretty complex architecture, and I'm happy to skip the details in this talk. Having talked about some of our work to utilize multi-omics data, I will switch the gear a little bit to talk about biological pathways and the networks. Genes and bacteria, they don't work alone. Genes, they form biological pathways, and bacteria, they may form communities. One of our early work is MinPath, which is a method we developed for conservative but hopefully more reliable inference of biological pathways from a metagenome. More recently, we developed a method called PhyloMint to quantify the potential metabolic interactions between bacterial species. When we say metabolic interactions, they can be competition or cooperation. Bacteria, they need to rely on some metabolites that they can produce themselves. So if we happen to have a species that can produce a metabolite that's needed by another species, there is a potential cooperation between these two species. And of course, if two species they compete for the same resource of metabolites, there's a potential competition. And if two bacterial species are phylogenetically close by nature, their biological pathways will be more similar to each other, and it looks like artificially they form a competitive relationship. But we need to adjust that phylogenetic impact on those quantifications. So we propose this phylogenetic adjusted quantification of metabolic interactions between microbial species, and from there we can identify pairs of species that, for example, metabolically cooperate, and from there we can identify communities of bacterial species. And for example, here on the left, I show a small community that exclusively contains bifidobacterial species, including bifidobacterium, longum, and infantides. Hopefully, those communities will give us some idea about some aspects of the functionality of those microbial communities. Another work we worked on is to infer communities of bacterial species based on the co-occurrence information. Thanks to the accumulation of these genomes and the metagenome assembled genomes, we have also a large collection of metagenomic data sets, so we can do abundance quantification of those genomes across thousands of samples. From there, we can infer the co-occurrence relationship between bacterial species. And also we took advantage of the recent advances of in-network inference that can deal with the sparse compositional data. We can infer those networks of co-occurrence network of bacterial species. And we can identify some of those network associated features that can help stratify between the healthy and the diseased states. For example, the differentiation of the various bacterial phylum, and also the enrichment of some of those interactions in disease network involving, for example, those protein or bacteria. Lastly, I will spend a few minutes to introduce some of our work related to CRISPR-Cas systems, the bacterial immune systems. We used CRISPR-Cas systems to study the species heterogeneity and the bacteria-virus interactions in microbial communities. Bacteria have many different defense mechanisms to fight against invaders such as viruses and plasmids, and CRISPR-Cas systems is one of those defense mechanisms. The CRISPR-Cas systems in bacterial genomes are composed of Cas genes and CRISPR arrays. And typically the genes and CRISPR arrays are adjacent in the genomes. Each CRISPR array consists of a series of direct repeats separated by unique spacer sequences acquired from invading genetic elements. In other words, the spacers found in the CRISPR arrays record the identity of the invaders, and the ordering of the spacers in the CRISPR array record the invasion history. Due to the constant arms race between bacteria and invaders, bacteria need to acquire new spacers from invaders, forming different spacer CRISPR arrays. By studying the variations of the CRISPR arrays in microbiome, we can learn about the interactions between bacteria and the viruses. The plot on the right shows the different stages involved in the CRISPR-Cas systems, the acquisition of new spacer sequences, the CRISPR RNA biogenesis, and the target interference against invaders, in which the mature CRISPR RNAs direct Cas proteins to silence complementary invaders. There are many types of CRISPR-Cas systems grouped into classes, class 1 and 2. And all those CRISPR-Cas systems contain some universal Cas genes, such as Cas1 and Cas2. The most famous type of CRISPR-Cas is the type 2, which contains Cas9 gene. It was the first CRISPR-Cas systems that was repurposed for genome editing and other applications. Due to the repetitive nature of the CRISPR arrays, they may be difficult to assemble from short reads. You can see a toy CRISPR array at the bottom of the slide. The array contains 3 spacers showing different colors, separated by 4 repeats shown in green. Nevertheless, we have developed some computational tools for assembling and the characterization of the CRISPR arrays from short metagenomic sequences. Once CRISPR arrays are characterized, we can extract spacers from the arrays and use those spacers to identify potential invaders. and from there we can study the interactions between invaders and the bacteria. As you can see from the bottom, the repeats on the spacers in CRISPR arrays are very long. So if we have long reads, we don't even need to assemble the reads to study the CRISPR arrays. As demonstration of this idea in this work, we show the promise of using long reads to study the heterogeneity of CRISPR arrays. We used the Illumina's TrueSeq synthetic long reads from a gut microbiome for this study, but the same analysis can be done using other more recent long reads, such as the PacBio HiFi reads. We show that long reads captured CRISPR spacers at a much higher degree of redundancy, resulting in better characterization of CRISPR variants, enabling the study of CRISPR arrays dynamics in ways difficult to achieve through short-read sequences. We introduce spacer graphs, a visual abstraction of spacer sharing CRISPR arrays to provide a simplified view of complex organizational structures present within CRISPR array dynamics. As an example, we show the variants of CRISPR array associated with a type 2 CRISPR-Cas system, as the plots on the left show, and the plots on the right shows a diversification of CRISPR's very extensive spacer gains. The nodes in blue are the new spacers inserted into the spacer sharing CRISPR arrays, forming many different ones, all found in the same microbiome. And the nodes in red represent spacers which we can trace back to potential invaders. Again, those invaders are found in the same microbiome. And the advantage of using CRISPR arrays to study this heterogeneity of bacterial species is that it's difficult to mess up with a spacer, a whole segment of 20 or 30 something bases. SNPs, on the other hand, if we use SNPs to study this heterogeneity, there may be mistakes because SNPs could be caused by sequencing errors. In this work, we used about 600 of those time-resolved genomes from Bacteroidetes fragilis isolates from multiple individuals and some involving multiple times to study the intersubject and intrasubject heterogeneity of B. fragilis. The plots on this slide show the spacer graph representation and also pie-up view of the CRISPR arrays found in B. fragilis at different time points of a specific individual subject called subject S06. The plots reveal that there is a major isolate of the B. fragilis which has the CRISPR arrays contain the spacers without any colors, and this isolate is shared by B. fragilis isolates found in different time points, and there are two different variants of B. fragilis containing CRISPR arrays with green spacers. The two variants share spacers 1, 2, 3, 4, 5, and one variant contains spacer 10, and the other one contains spacers 6, 7, 8, 9, and 11. We can also see variations of this CRISPR arrays with deletions of spacers. We can use spacers found in CRISPR arrays to identify potential invaders of the various bacterial species in microbiome. As an example, in this work, we analyzed 5 water microbiomes. We characterized the species composition of the water microbiomes using shotgun metagenomic sequences, and the plot on the left shows the species composition of the 5 water microbiome. We used we characterized the CRISPR arrays, and from there we identify spacers, and using the spacers we identify all the potential invaders through the spacers. From there we can build an interaction network between the microbiome and potential invaders. And the interaction network shown on the right suggests that the 5 microbiome have very different histories of virus invading. For example, if we look at one of the microbiome 3 and 5, they do share the most similar species composition, but if we look at the interaction network, we see these two one of the microbiomes don't share a whole lot of invaders. We have seen recent advances in computational tool development for analyzing microbiome data, including some of the tools we have developed and that I showed in this talk. But there are still many microbiome data sets out there, and many are underutilized. So if you have the right questions and then you have the right tools, or you can develop the right tools, there will be great potential out there. And with that, I would like to thank all the students in my group who have worked on the various microbiome projects and my collaborators. And I would like to thank you for your patience.